Thank you all for being here this afternoon, and it's a great pleasure to introduce Gabrielle, whom we've all heard a lot about, those of us who've worked on projects with Nisha and Ian. And Gabrielle is coming to us from the Institute de Cardiologie, where she is as part of a lab led by Dr. Jean Rioux. And unlike our premises here, where many people are in clinical research, Gabrielle works more with biologists and with clinicians, and she's been there for the last 14 years.
Yes.
And interestingly, Gabrielle has a number of observations about experimental study design, something that's often ignored when people are carrying out statistical analyses. And so today she's going to share with us some of her learning experiences over the years about meeting a statistician upfront, rather than when it's too late.
Yeah.
Okay. Thank you.
So thanks, Nandini. So as Nandini explained, I'm working in a molecular biology lab. So I'm the statistician of the lab, and I started just fresh from university, knowing a lot of stuff about statistics, but nothing about my molecular biology. I knew about genetics, but not about what they do in the lab. And for the last years, I learned a lot from the people I work with, because their reality is really important to know when we need to design a study and to analyze the study. This presentation I first did for biologists. I adapted for today. And okay, so just by the look, I will know if you seem to understand or not. So the first thing is the feeling I had when I started working with biologists as a statistician is that it feels like we're supposed to be superhero, but we're not, and we know it. And this is StatMan, but StatMan does not exist in real life. And this is a creation by a friend of us from university. And we can help with the design and to identify problem, correct some issues, create plots and those sort of things. But sometimes what I have as a feeling is that people come to us asking about saving an experiment because, okay, I did this experiment and something went wrong, and can you save this for me? So this is why I learned that the most important step for optimal statistical analysis start at the design stage. And statistician will learn about statistical design, but this is not what I will be talking about, because statistical design, like, for example, factorial design and those kind of stuff, we never do really that kind of things in molecular biology because there's so many things that can go wrong. It's mostly about really how the study will be done, how the experimental thing will be done. So as I said, the design is the foundation of the experiment, and if the design is wrong, everything will go wrong. And what I learned is that many biology students, they learn how to do t-tests, for example, but they never really learn how to do proper design. And they come to us after the fact, and we can always help them do the analysis, but we can't never go back and redo their experiment when it's not done properly. So I like to sometimes remind people that statistics is not only about numbers and formula, because people come to us and say, oh, I'm not good in math, so just help me, thank God. No, it's much more, I think, about inference, logical rules, problem solving, and we use maths to do that. And it's about what we can say with the information we have and what information we need to answer a question. So when a biologist or a researcher come to me, I'm telling them, what do you want to do? What is your question? What is your your biological question? And then I try to help them. Sometimes people come to me and ask, okay, I want to do an ANOVA, and then, okay, you don't want to do an ANOVA. This is not what you want to do. What you want to do is you have a question, you have data, and then you want an answer. Maybe an ANOVA will be the proper way of doing it, but it's not your objective to do an ANOVA. So this is a funny example that I sometimes tell people. Yeah, some people here in the room. So this is an example that I come along working in the lab to explain something about a very common design problem I see in the lab. So let's start with a question. Are men taller than women? And then I have men, I have women, I sampled a few people, and I want to measure them, but I'm not very good at measuring people, so I measure them 10 times. So I will have technical replicates. So I will have a very precise estimate for these exact 6 people. So I will be able to conclude with certainty about those 6 people, of course. But I will still have N of 3 to compare the groups. That's 60. So people will come, I have 60 data points. Yes, sure, but you have 6% here in your data set. And then they will say, okay, let's redo the whole experiment with the same people. I'd say, okay, it's the same N of 3. Of course, this is not exactly what we do in the lab. But what they do in the lab is they redo the same experiment with the same cells over and over and over again. They do the same experiment with the same rats over and over and over again. Or they do different kind of experiments with the exact same rats. So they measure different things, but it's always the same people. It's always the same rats. So that's, but they still have just N of 3. And we need to explain to them this fact. It's important. So when I started in the lab, as I said, I knew how to do stats, but people came to me and saying, okay, I have those biological replicate, I have experimental replicate, I have technical replicate, and how should I deal with those? And I didn't even know what were those concepts. So the biological, for people working in the lab, it's at the level of the individual or the biological sample. The experimental is at the level of the treatment. You treat the cell, for example. Well, the technical will be at the level of measure. So I treated cells and then I measure something X amount of time. So it's not always as simple to define. It depends on the experiment. It's not always that important. What is really most important for statistician is what is the experimental unit and which population we are talking about, and at which level of inference we are. So if I have, this is a common experience in immunology, for example. So I have Phil, and Phil, we took blood, and then from the blood, we isolated cells. I don't do that. Of course, I don't. So we isolated cells, and then we froze those cells in small tubes to keep for later experiment. And then we take one of the tubes, we thaw the tubes, and then we use the cells and the cells in different wells. And then we decide this well will be treated, the other one will not be treated, and we want to compare that. Maybe we will repeat the measure on the wells. So this is what we have. So the biological, the experimental, and the technical here. But what we need to understand is that while the technical replicate are there to increase the precision on measure, because sometimes there is a lot of noise in what we try to measure, like when I'm not very good at measuring people. The experimental unit here, we can consider it to be the well, because it's a well that we decide to treat or not treat. But if we do that, then it means that we can only do inference on the tube that we thaw, because maybe the tube, something happened to it, and it's not even similar to the other tubes of cells. And maybe those tubes of cells will be or not really representative of the individual, because maybe on that day that person was sick or not. And is Phil really representative of the whole population as for himself? So we have only N of 1 for the population, in fact. And this is a very common study design, and sometimes we will increase the number of people, but this is really something that we do often. So what I'm trying to explain to people in the lab and doing those kind of experiments is that it's usually best to maximize the number of biological samples. And then if there's steps in the experiment that is really heterogeneous, or there's noisy steps, because it happens, then we need repeats. And this is the step where we need the repeats. For example, in the lab, we sometimes take, for example, skin cells, and then they reprogram those cells so that they think they are stem cells. And then they treat those cells so that they think that they are small guts, like baby guts. But this whole experiment is really noisy. So this step need to be done a few times so that we can say something from the data we will collect. And if the experiment process is really more noisy than individual variation, I tell them, you need to talk to a statistician because you will need to have a trade-off in your experiment. And one thing, because I work with biologists, I also work with clinicians. And one thing that I saw a lot is case control designs with human samples. I mostly work on genetics, but I also work on lipidomics, proteomics. And then what will happen if we have cases and controls? So we have diseased people and not diseased. That's look like that's all we need. But it's not the case because we need them to be from the same population. And it's not always the case. So I was working on that study where they wanted to look at some cardiovascular disease, which happens later in life with mean age about 70, I think, if I remember well, and mostly men. But then they recruited, and they needed to recruit them, and they were really specific about how to recruit the people that were sick. But then they needed control. So they took whoever entered the clinic. And they even non-sick people, staff people, etc. And they ended up with controls that were about 40 years old and mostly females. And we wanted to do metabolomics, and metabolomics is really affected by age and sex. So it was a problem here. So we needed to, we found a way to correct for that because there was a lot of controls. We ended up removing a lot of them, and the sample size shrank a lot. The other thing that I've seen a lot is use of biobank. So usually biobank is within an hospital, and people are just collecting a lot of people, where they diseased people or staff people or family members of of patients. And it can really be useful. It's a great resource because there's a lot of people in there and a lot of data in there. We have one at the Montreal Art. So the problem is how and why were they recruited? There's a reason why the people are in the database. And sometimes they are more healthy than the controls you will need. Sometimes they are more sick, and sometimes it's even worse. We can end up comparing two diseases. And this is something that happened to us. We were looking into the biobank, and this was a cardiovascular disease, and it needed echocardiography, and then to be diagnosed. Yeah. So we selected the case to be positive at this exam, and the controls to be negative at the exam. So fine. And then we compared them, and we realized somehow that the controls had another type of disease, because there's a reason why you will have an echocardiography. I never had one. I don't know for you, but I never. So this is really important because this can create artifacts. Or what will mostly happen is that you won't be maybe answering the question you had in the beginning. You will be answering another question. So what I tell people is that the selection of control is as important as the selection of cases, and maybe even more. So this question here, do you have gastrointestinal fistula? So for most people, the answer will be, I don't know. Why? Because this is highly specific. This is something I work mostly on Crohn's disease and ulcerative colitis. So this is a complication of Crohn's disease, and a very severe one where there's, sorry for the the not medical explanation, but like perforation of the gut that will make the organs to connect together somehow. And this is really painful. So if you have one, chances are you know it. Chances are your clinician told you and you know it. But if you don't have one, you won't know, because you don't even know what it is. And then we add this large cohort with many different groups from different hospitals in the world. And one of the cohort, they had such a high rate of this complication, almost if everyone had it, but it's about a third of the patient who will get fistula. And what we saw is that many of them, there was also a very high rate of unknown. And then what we realized is that this specific cohort, the questionnaire were filled by the patients. So the patients who had a fistula, they knew it. So they said yes. And the other one, some of them said no or unknown. So we had to completely exclude that big cohort due to that. And this is non-random missingness due to study design, because if it was a clinician who had filled the paperwork here, the questionnaire, the clinician know if the clinician know what is a fistula. So yeah. No, because there are real unknowns. Because there are some of the patients for which this was a genetic cohort here. And for some of the patients, we just didn't have the information at all about what we call disease behavior here. So is it true unknown, or is it unknown due to the fact that the person didn't know? Sometimes we know it because it's really unknown or missing. But when we collect cohorts from everywhere in the world, you end up with 30,000 people. You somehow collapse those. You need to simplify this. And the other point is, some patient will still answer no, some patient will still answer unknown, and you can't always completely, you can think that probably it will be that, but it's not perfect. So we just excluded that one. But yes, for some other context, it could have been, but it will have been much better if just the clinician filled the questionnaire. So also, again, about some non-random missingness. I don't know if you heard about this example. So during World War II, planes came back with bullet holes, and they were wondering, can we put some plates on the plane to protect them? So they looked at the data and observed that they were where the holes were. And they thought, okay, so let's put plates where there are a lot of holes, of bullet holes. But then the statistician came and he said, well, where are the missing bullet holes? What about the planes that we can't observe because they never came back? Okay, they never came back, so we can't observe them. And when can we think that maybe the bullet holes will be is in the cockpit? So if the plane is shot in the cockpit, the plane crashed and we never observed this. And this is the worst scenario of missing value, because you don't even know that you don't know. And study design can sometimes help us do that. An example that, I don't know if you knew, there was some kind of pandemic, and then there was many different studies that came, and for statisticians, it was a nightmare reading everything that came out. So this study here, they wanted to investigate smoking. And there was already different studies that came out about smoking and the risk for people who smoke to get very severe disease. But they came to a different conclusion, that smoking was protective. So We sat at the dinner table and we read the paper. And it's all started with, okay, they took patient admitted in the hospital. And they compared the smoking rate between the patients and the general population. And they said, well, there's less smokers in the patient in the hospital than in the general population. So they said, smoking is protective. And then, at the very small details at the end of the paper, they're talking about limitations. So limitation, they excluded all the people in intensive care and all the deaf people. And it was already known by other studies that there were more smokers in those very severe cases. So their limitation wasn't a limitation, it was a major flaw that completely invalidated the study. So this was a preprint, so I don't know if it was, I never came back to see if it was published at the end. Probably didn't do Lensa. Sorry? I have the paper somewhere on my computer. It was a preprint, and yeah. So yeah. It's a major problem because this was published as a preprint, and then the press, they took it, and then everybody was talking about it. Okay. Yeah, you need it to be sub, and that's why that's why I'm saying I never came back to see if it was actually published, at least in a real paper, in a real journal, and not one of those that you need that just are peer reviewed and real peer reviewed, but that's another subject. But the design, this is still the design that they came about with. So, and this is something that can happen in studies. Of course, I will never have accepted that at the review, but I mean, so now let's talk about Murphy's Law. So we all know about this Murphy's Law, that if I just my toes fell on the floor, it will fall on the bad side. But what most people don't know, if at the beginning this was about experimental design and being sure that you won't kill the pilots during your study. Okay, so what they did, they considered every possible thing that could go wrong, so that the experiment at the end won't kill the pilot of the plane. But we need to do the same when we do a study design. And this is for lab, but you can think about clinical design also, that you will have played batch effects, side effect between different clinics, position, time trend, this happens sometimes. You will lose samples. This happens. And what will you do if you lose samples? So, for example, those very neat factorial design, what will you do if you lose a few samples? And it probably will happen. Equipment failure and anything that could go wrong. And then we need to protect the experiment about those problems, so that at the end, all the work and resources that were put into the experiment will be protected. So this is at the design stage. So what I tell people is to always consider that there will be played batch, side effect, those kind of things. And those are systematic difference in this distribution between batches, for example. That never trust that you can avoid it or correct it. Because sometimes, for example, we had this Luminex platform, and people, the people from Luminex, from the company, they told us, oh, there's no plate effect once you put standard curve. But what we found out that it's not really true. It's true only for a very small range within the curve, but for the whole range, it's not true. So just don't trust that. So when I was a baby statistician in the lab, so this student came and she learned about study design, and she told me, okay, I have this experiment. I have a 96 plate where I need to put my samples, and then I need to treat my samples with different treatment. And I know that there will be border effect. Whoops, sorry. I know that there will be border effect due to evaporation of the samples, and that there could be also other position effect like rows or columns, etc. And what I told her, well, no problem, just randomize, just randomize everything. And then she looked at me like I was completely crazy. And she told me, okay, you don't know what it means, because if I need to randomize, it means that I need to put every single sample with a one channel pipette. And then I need also to put every reactive like that. And then I will mix up everything, because there's nothing less similar to a small transparent tubes and another small transparent tube. So that's how I learned, because they have those cool pipette were multi-channel, and then okay. So okay, then what I learned is it's best to then balance samples between plates, the batches, and break pattern as much as possible. So, and we had a robot in the lab, and we figured out that the top row, it was always higher values for whatever reason. So if you always put your cases on the top rows and your controls on the bottom, you will have systematic bias here. This is one kind of pattern that is very common in molecular biology, is the 96 well plate with two columns being the standards. This is serial dilution. And we put the samples, and then we compute the standard curve. But there usually still are plate effects. This is a standard curve, one type of standard curve. This one is sigmoid. There are more simple ones. The simpler standard curve is just linear, but most of the time it's not the case. So you have this standard curve. And what we do is, again, not me, but we start with a sample with usually with non-concentration, and then we do a first dilution, and then from this one we do a second, and this is serial like that. And we put that on the plate. And then you get the concentration here on the bottom, the fluorescence. Here it was a study with fluorescence as a reading. And then we compute a standard curve, which is a 5PL regression, which is 5-parameter logistic, where at the bottom you have the lower limit, the upper limit, and then you have a parameter for the steepness of the curve here, a parameter for something about the middle, and another one for asymmetry between those two inflection points. And these kind of curve, the fit is really sensitive to just even a small deviation, a small random noise at the extremes, in particular at the bottom of the curve. Because at the bottom, you're close to the limit of the machine, of the equipment, but also you did serial dilution. So if there was an error somewhere, it will accumulate to large error at the bottom. The numbers here are the recovery. So we prefer it to be 100%, which is when we compare the concentration that we estimate from the curve to the real one. And we consider that we have good data when we're close to about 90 or 80, and then the rest is extrapolation. So this is an example. We have 4 plates. The standard are named with S, and the samples are the numbers at the bottom. And this is the fluorescence, and we can see there's some differences. And then we compute standard curve for each of the plates, and this is what we get as a concentration for the different plate. There are obvious plate effect, very large difference. And the most dramatic one is this one, where for some reason, something happened with the curve here, and all the sample fell below the threshold of detection. So what I did is I did some magical statistical stuff, and I corrected the fluorescence to make it more similar, because it was a balanced study. And then I compute a single standard curve, and everything was a bit more similar, but there was still some plate effect even there. Yes. because I know that each of my plates have a similar breakdown of the different type of samples, so that they should have, on average, a similar distribution. They should, because this is how I created the plates. But I couldn't do that here if it wasn't for the plate to be balanced. So what I usually tell people is to keep samples they want to compare close together to avoid artifacts such as case control, treated, untreated, and close together being the space on the plates, time or batch or reactive or anything like that. And then I prefer to have minimal distance between the biological samples so that we reduce sample noise and then increase power. And then the technical replicates can usually be confounded with batches. So if you want to do it twice, then do two plates. Well, it's not a big problem unless you are interested actually in technical replicates. And when limited by resources, you need to do multiple batch to balance and randomize. So the second part here is very short one. I shortened it just to show what I think is really important and that many people, even statisticians working in biology, don't know. So most people know that the accumulation of random additive effect will usually yield to the famous normal distribution or something that will be similar to normal or Gaussian distribution. This is what we learn when we do stat classes. But the accumulation of random multiplicative effect will give us a log normal distribution. And this just follow from one another. And many distribution in biology are log normal or close to symmetric log normal like, because many effects are multiplicative. And in fact, there's a mixture of both type of effect. So the log normal, if we log transform, we get back the normal distribution. This is where the name comes from. And this is why we log transform data in biology. This is the reason why we do that. And then what it will be multiplicative effect on the original scale, we become additive on the log scale. This comes from the law of logarithms that we all learned in high school and thought that we never used again in our lives. And you can come back doing the reverse transformation to get back after you did your stats here, after the transformation, you can go back doing the reverse, and then you get effect on as a fold change, and you can get an estimate of the median being the geometric mean. And this is what I almost always do. So when I look at data that comes from those kind of experiment, I start with log transformation, and then if log transformation do not seem like appropriate, then I go back to other type of analysis. And these are some hints that you may have a log normal distribution. Biology is a good one. The process is known to be multiplicative like cell division. So cell division, of course, it will multiply by itself. When we measure something in fluorescence units, it usually is. The measure will be strictly positive because you multiply the result. You already think of the effect in fold change, and that's happened a lot. So people in biology, they come and, oh, I expect an effect to be a twofold. I expect the effect to be an increase of 50%. So that's multiplicative effect. The variance scales with the mean. So if you have different condition and the bar plots, because we always do bar plots in biology, the variance scales with the mean, it probably is multiplicative. Or a very skewed distribution, or for statistician, a V-shaped residual plot. What I've seen a lot working in biology is this question, which p-value threshold I should use. And often I tell people, maybe, maybe. because for many students and some researcher, p-value is the only way to go to do statistics. I know it's not the case for some people here, but in my field, I've seen that a lot. So what I recall people is that statistical testing is a special type of statistical inference, and it's not the only one. And we usually test for no effect, no difference. And then the type 1 error, which will be your p-value, is the accepted risk of false positives. So it's up to the researcher to decide what is the accepted risk of false positive. And it will also always be a trade-off between the cost of a false positive versus the cost of a false negative. And it's up to the researcher to decide that. And sometimes when H0, when the null is no difference, well, sometimes a false negative is worse. For example, if you're looking into adverse effect of a treatment, it would be worse to just to just accept false negative and say, oh, there's no problem with my treatment. But when people are actually dying, in those situations, we can change H0, but it's still the most common one is no effect. And then I'm telling them also, are you even asking asking the right question? Because I found many people are just doing, okay, let's do a statistical test. If my p-value is significant, then I will look if the effect is greater than X. And then I will have a result. So then I tell them, okay, so then your question is not whether or not there's an effect, but how large is your effect? Is it larger than X? Sometimes we also even know that there's an effect. I've seen that they know there will be an effect because, for example, it's a metabolic disease, and they're doing measuring metabolites affected by the disease. But what they are interested in is the scale of the effect. But they still do a statistical test and look at p-value. So I tell people, well, to focus more on the estimation of parameters and errors, and to do really plot like this, and maybe draw confidence interval. And this, I saw this a lot in presentation from biologists, from students, is they do a statistical test, the p-value is non-significant, and they say, well, there's no difference between the treated and untreated, and the data looks like this. And I would say, well, I'm not quite sure. It's not significant, but well, it looks like maybe there's something here, but you're just lacking information. So I usually tell people, why don't you use confidence interval here? Because then you will be able to say the difference between my effect is smaller than X, like this. The confidence interval is close to 0, so no effect. Or there's a lack of information, being my confidence interval is just Y, telling me that I just don't know. And this is not the same, but your p-value will make the difference between those two situations. And this is done a lot in presentation. So this is the second part of this presentation, and this is one project we have in the lab. An ongoing project on inflammatory bowel disease, Crohn's disease, ulcerative colitis. Those are diseases that affect the gut. So Crohn's disease is the whole digestive tract, while ulcerative colitis is really the colon that is affected. We were implicated in genetic association studies. We have a lot of genetic variants that are associated. And now we try to understand the biology underlying those genetic variation by doing experiments in the lab. And also we are looking into treatment for IBD. So there's heterogeneity in IBD. For the localization, being the different part of the gut that can be affected, but also in the behavior being just mild disease where the people don't feel well, to extremely severe where the gut can block or create fistula, and people will need to go through surgeries, and even sometimes they can die actually. And this is those are diseases that people usually are diagnosed in their 20s or 30s. So they will be sick for a very long time. There's also heterogeneity in the response to therapy. So my disease, usually they will use AINES, they use steroids sometimes, or other kind of treatment. And when it fails and people get more severe disease, now they are more into new classes of medication, being the biologics treatment, which are mostly antibodies or those kind of things. And those works very well for some patients, but not at all for others. So we're trying to say, is there a way to measure different parameters, different biomarkers in the patient so that we can say, let's start by trying anti-TNF, or let's start with trying anti-integrin instead. Not saying you won't respond, but just maybe the pathway that underlies your disease will respond better to one of those treatments. So it all started a few years ago, and we did some experiment. We still do others. Some are already published, and now we're at the stage of getting prospective cohorts to do that. One day we expect to get a predictive score. One day. So what we do, this part, the first phase, was done in Montreal. Sample recruited in McGill University Center and other at the CHUM. And we took samples at baseline. We took also samples at 16 weeks and other at 16 weeks post-treatment and after 1 year. One of the things that we look at is supernatant after stimulation. So we took the blood, put them into different tubes with stuff reactive in the tubes to stimulate the cells in the blood. And then we wait for 24 hours and we harvest the liquid at the top of the tubes. And then we freeze them in small tubes. And those stimulation, they will stimulate different specific immune cells, and they usually have huge effects. So we also collect clinical data, and this is important. We have some of the reports will be filled by the clinician and other with a patient, with a research nurse with them to help them understand the CRF. So we collect a lot of information. Then the blood is collected, sent to the Montreal our Heart Institute, where we extract the serum, the supernatant, and we freeze them and keep them in the -80 freezer for it was, I think, 3 years in total for the recruitment. Of course, we would have preferred to be able to get 200 patients on the same day at the same center. It would have been very great for a study design, but it seems like it wasn't possible. So this is how we did it. So what I will be talking about is this first phase with a bit less than 200 patients. We have serum proteomics, for example. I will talk about those, and supernatant. I will also just talk very quickly. And we decided to measure, we came with Olink, and we measure cytokines and chemokines, which are protein that's in signaling between the cells. And then before doing the design of this part of the study, we met with the expert of the technology from Olink, and we asked them, how do you proceed with the samples? What do you do with them? Because we weren't doing it in the lab. So we asked them, and they told us that they run those 96 well plate. They use one column for controls, and all the readings are done at once. This is important because it reduces plate effect. If the reading was done by columns or by row, or even Luminex, for example, I don't know if they don't go like this, though, so it's run across the plate. So it's important for the design. So how do we place the sample on the plate? So this one was quite simple. So we needed 3 plates or 2.5. And I decided to balance for sex, medication, age, and visit between half plates so that it will be about similar all over the place. And then I put some repeats between the plates. And the few samples for which we had post-treatment samples, I also just randomly placed them on the plate, close to the other at baseline. And this way we can compare them. We have few samples after treatment. For practical reason, we needed to stop the recruitment of those, the collection of those. And then the supernatant, I just place them like this. So the color is the stimulation. If you observe here, I do have a pattern here in my plate, but here for our situation, it wasn't a very big problem because the stimulation is expected to have huge effect, much more larger than any possible plate or position effect. So if it was an issue, we could have broken the pattern like this, but it wasn't a problem for us here. Then the company did the measure, they identified failed samples, the limit of detection, and they did actually the plate normalization to reduce the plate effect. And they could do that because the study design was balanced. And this is something that they told us, we need a balanced design. And they sent us a quality control report and data. So the next step, then, once I have my data, is always what I always do is I explore the data and I try to find problems. So I usually take a few weeks at trying to find problems and investigate the reliable analytes. One example, and this is why I put replicates between the sample, the plates, is this analyte, the replicates correlates very well. So on the x-axis is replicate 1, y-axis replicate 2, and they fall quite well on the line. So this is a good quality analyte that will be easy to analyze. This one here, it's almost random noise. So what it means that is the data is not really usable for any possible interpretation. Then I do PCA. I suppose most people here know what it is, but it is a 2D representation of my sample. And then I say, oh, there's a problem here. I have this bunch of samples that do not behave like the other ones. But then I figured out that it was a phenotype of the samples that we identify here. So this is a true signal. But I always first think that it's a problem. This is always my first reflex. And for the supernatant, I also did a PCA, and we can see that the different conditions separate quite well. I have a 3D PCA, but I don't think I will be able to show it now. And it's really cool because we can really see the groups separating. But I have a few dots that do not belong where they are. And then I investigated and found out that, oh, these were mislabeled samples. So this is the first step. So next, what I'm doing now is the proper analysis of the data. It's not an easy step, but it's for later on. So in conclusion here is that life science experiments often face issues. And there's nothing that we can do about it. There will always be some kind of problem like mislabeling of samples, plate effect, losing data, someone drop sample. We had one that we were waiting for during the ice storm. What can we do about that? So the planification is key to make sure that we can still interpret the data. And expertise in technology and inference like statistic is important to do that. We need both. Just statistician alone, you can't do it. Just biologists, it might be tough if you don't have a proper statistical training. And what I found out is that often students in biology, they got a statistical training that is not really related to what they will do in laboratory, in the lab. So they end up in the lab and they're not sure what to do and how to use the stats that they learned in the lab. So probably there need to be a better bridge between the two for biology student and even for biostatistician to really understand how it works.
Question, Dr. Bernasky.
Yeah.
How did you figure out the sample was mislabeled?
So it took me a whole week to do that. And so one slide, but yeah, so for some of them, I just figured out that there was a problem. I correlated each of the samples. I know the patient, the subject name, and I know the condition it's supposed to be. And then I know some of those stimulation, what they should affect. So cytokines that should be affected by LPS. For example, LPS is like putting a bomb into the cells. So I know that. And then for some of them, it was obvious because it was the same subject ID, but it was just the two that were like, okay, inverse. So it was easy. For the other one, I needed to really correlate them with the other conditions. And what I did is I removed those for which it wasn't obvious that it really was a swapping of samples. I just removed them.
Congratulate you on your detective work.
Thank you. I think my work is a lot of detective work. Yeah. Thank you. Yes.
That would made us nervous because in epidemiology, of course, we're dealing with, we have our data and they've all undergone assessments, and increasingly some of the assessments are biological tests that are maybe a bit more complex than the tests that we're used to. And we were happy to say those things were taken care of, and our only problems were our blood pressure measurements, our measurements. Well, we recently, who I think has been trained by you to look for badge.
Not trained by me, but
But not with the badge effect.
Yeah.
But it was thinking, you know, like it's interesting. I suspect often these things are missed. And
yeah, I think one thing that happens is that the people doing these kind of work in the lab, if I go back to the standard curve, I'm sorry because I can't control the presentation from just a slide. I just want to go back here. What they learned, it's not really their fault. I mean, they really are told by the company that once they put the standard, it will be okay. But it's true and it's false. So it will be true in that range here. In a very small range of very good quantification, if there's no issue doing the standard curve here, I mean, but it will be quite true because this part here, there's a lot of information for quite a linear trend. So it's quite good, but often and ideally we will go with samples that will fall here. But in a real life situation, it happens that samples fall here, and this part of the curve is really not reliable. Yes.
One of the things I've learned, I've been taught as well, is from your beginning, like the slide at the beginning, is to have repeated, like to repeat multiple biological samples and just sort of repeat, right? Repeat the technical.
Like this, for example.
As many as I can. So, for example, I'll take from Phil and his family and his friends, and then take like for the two of them, take them separate, then do them into different separate wells, and then repeat that twice. But then, and I mean, would you say that would be the best way, or there's a better way? Because there are always people who would think, why should I bother? If it worked the first time or with the first two repeats, and just like you said, right, repeating with the same people
over and over again.
That's representative there, that this is what the effect was, which is the best representative from the technical sample. So what would you say to either way of them? Like, is it good to have that piece of multiple repeats of the biological and then repeat as many as possible, even though, as you showed later, there are different effects of the plate, of course.
Yeah.
If you're getting say 5 times.
Yeah, but it's a complex question because it really depends on the experiment. I would say there are different stages when we do experiment like these. The first stage in the lab is always, well, we're trying to understand the assay and how variable it is. Then we don't really care that much. We'll have a few people, maybe 3 or 4, and do repeats, as you're saying. And we try to identify where is the most variable step. For example, in FACS data, where you try to identify cell population, this is really noisy data. It can be really affected by many different things, and just the towing, just this tube can be completely different from the other one as you tell. And then in that situation, I would say you need a lot of repeat at this stage here. So you need them, but then you will not be able to do all of your biological sample at the same time. So what you will need to do is, for example, you will have different people, different tube, and then you can have fill 1 tube, another people 1 tube, and then you redo the experiment. And next day it will be the same people, different tubes, and then you redo it. Yeah.
So basically, but then depending where they're, and then the best thing to start with is having enough biological sample, which is more than 1 pill.
Yeah. And there's someone in the lab actually. but it really depends. I mean, if you're really into just understanding the technique, it's not the same thing as if you want to say something about the population.
I think Ben had a question.
But I was curious.
Thank you.
Find the trajectory of science towards team science and atomization of the actual experiment, where different parts are being done by different people on different places. So, but show really nicely that you really need like exactly what's being done every step, all of the nuances. So operationally, how is your research group set up? It sounds like you must kind of in the lab almost saying like, wait, what did you do there? Like, wait, what was that?
Okay.
Like,
Yeah. Yes, I'm being told that I was kind of a pain for that. Yeah. So if we go back to, for example, the specific project we are looking at, I work in John Rieu's lab at the MHI, but we are part of larger consortium in USA and Canada. But this specific study there that I've shown. Okay, so this part here, it was all done in Montreal. We're not now going into further step with further stages where we will have people from all around Canada and USA. This will be next stage in the next few years. So what we do, we have we need to work with those people. I'm still kind of the main analyst now for this. So we have meeting every 2 weeks, and we talk with people, and the clinicians, lab people, statisticians, they all talk together, they meet, and they say, okay, well, we need to get the patients that way, and then we prepared, we talked about it. And then the clinicians say, well, no, I can't do that because, no. So, okay, then we'll rethink about that. So it's a for and go, and we always correct what we're doing like this, talking with the clinician like Alain here, Dr. Biton. So we talk with them. And then the people in the lab is the same. Okay, now that we have the samples, okay, what do you do? And then we talk together. But in the lab, we have me as a statistician. The other one are within our lab are molecular biologists and geneticians. and we work with clinicians that are outside of the lab. Did I answer your question?
It sounds like having regular is going to identify all the sources of variation. For having a statistician or the epidemiologist.
Yeah.
Go away.
Yeah.
Pick up on, oh, that's a source of their batch effect or whatever.
And also I just standardize it, standardize. Like all the workflow hours, for example, different centers as much as possible.
Yeah, one thing that we do here, and I just said it very quickly, is we have a centralized database. And by centralized database, I mean, we have the CRF and then Julie in the lab, she take each of them and then she enter the data into a database. If something is wrong, she go back and do a query to the nurse, correct that. And we work together to create the CRF and to create the database. And it's not some Excel spreadsheet. I mean, it's a formal database where you can't just mix everything by doing a wrong copy paste or filtering of the data. It's not that Excel is always bad with me.
Then point that we're working on a study on vaccines that you may have heard of the vaccine study, but it's very, very difficult to know, like you're working with labs in Ottawa, what exactly is happening there. And what we learned is that everything has to be done at the study design phase, that it's very difficult to control or change or get the answer you want at the statistical analysis phase. And you know, I don't think we've yet learned that we need to involve people, dermatologists at the study design phase, and it's also logistically very difficult.
Yeah.
Yeah. So we work mostly with FB studies. So I'm curious, in basic science studies, how often do you see people report in the methodology the batch effect? Because, as you said, we don't know what we don't know, but we unearthed it because we did that there was a batch effect that kind of invalidated the results. And so, okay, because of you. So how, you know, if we didn't know that, we get completely biased results. But I was curious to know whether basic science studies actually talk about that specific.
In our paper, we always state what we did to avoid batch effects. But I think there probably are many papers out there where people are not fully aware. And the problem is the review process of paper, it's great, but it's not perfect. If any of you did some review of a paper, you can't deal with every single aspect of the paper. I mean, it's complicated and you don't have access to the data, and something can easily slip under the carpet and you don't know it. For example, if I was to review a paper and I know that they have thousands of samples, I will ask, okay, how did you deal with the batch effect? Because I know that usually there's 96 samples on the plate, or sometimes more because there's those larger plates, but I will ask but you need to really be working on those kind of data to know that. So, and there's a thing is, I know there are a lot of constraints, money. So design is often, we need to do what we can with the time we have, with the money need to be spent now. We have the samples now and the freezer is full, so we need to do them now. And so sometimes the design is not the optimal statistical or inference design, but just practical-ish things that. But at the end, sometimes it's okay, but sometimes it's not.
Thank you very much, Gabrielle.
Thank you.
We'll be reaching